for that. And thanks everyone for coming today and the opportunity to talk about my research here while I'm visiting. Today I thought I'd split my talk into two parts. So the first half of the talk, I'm going to go over exactly the Red Sea project that he talked about, basically a big multidisciplinary project where our aim was to really catalog biodiversity on Red Sea reefs. It involved the Reef Ecology Lab, headed by Dr. Michael Bowen at the Abdullah University of Science and Technology, otherwise known as KAUST. There's a number of international collaborators and PIs, some of them are in this room today. And so it was a really interesting project, so I'll go through some of the outputs from that project, even though we're just starting to get to that stage now. The second half of the talk, I'm really going to focus on a lot of the things that I'm doing at Curtin University now. So I've been based there for just over a year, where we're trying to apply some of the environmental DNA methods to, I guess, the marine sector. And so that's some of the work that I'll focus on today as well. So before I kind of dive into the Red Sea project, I thought I'd outline some of the main research themes that I focus on, particularly since I'm new to Australia. And so you'll get a sense of why I'm interested in the questions that I'm asking through the rest of my talk. So the first main theme really focuses on looking at the origins of biodiversity. So where did things come from and when did they get there? Most of what we do uses molecular approaches. So whether we're looking at a single gene or a whole bunch of genes, we usually do this across a large number of individuals, large numbers of taxa. But we also pair this with morphological information, which is important as well. So with those two pieces of information, we can start to try to get at estimates of things like species diversity. We can resolve some of the taxonomic uncertainty, and also look at the evolutionary history of some of the more complex groups. The second theme is really focused on the maintenance of biodiversity. So what happens once something establishes in a new location? This is really related to the fields of phylogeography, population genetics, how much or how little different species or populations are connected to each other in terms of exchanging reproductively viable individuals. For a lot of these types of questions, we've really made the push towards some newer genomic approaches. And these are kind of nice because you can then overlay them with environmental models that, so you can look at how they interact with sea surface temperature, salinity, nutrient load, and so on. So you can really start to look at drivers of connectivity as well as patterns of connectivity in natural systems. So kind of a summary of the Red Sea project. It all started at KAUST. For those of you that aren't familiar with KAUST, that's okay, it's a pretty new school. It was established in the fall of 2009, I believe. It basically hosted local Saudi graduate students and a whole bunch of international graduate students as well. So you can see this is where KAUST is located, kind of on the central Red Sea coast. It's also strategically located in that there's a small little boat harbor, research vessels, so basically you have immediate access to Red Sea reefs. And so it was a great launching point for these projects. And so basically, a lot of the individual professors coming into KAUST at the very beginning, they called it the KAUST experiment because they didn't know how it was going to end up. But they basically saw that there was a real lack of available information from the region. And so this set the scene for them to be able to provide kind of this critical baseline information for the Red Sea. And just to show you kind of the lack of information, or at least available information from the Red Sea, this is the number of publications through time on a particularly hot topic in marine science, marine protected areas. And so the green line represents studies in the Caribbean, the red's Red Sea, and then the blue is the GBR. Clearly the Red Sea is way behind, although in terms of physical size, they're probably comparable. In terms of biodiversity, the Red Sea is way more diverse than the Caribbean, and it's probably somewhat on par with the GBR. And if we extend that search to a whole host of other areas or topics in marine science, so things like studies on apex predators, coral bleaching, connectivity, so things that I'm interested in, you can see that this lack of knowledge is really pervasive in all these different research aspects. So basically, one way that we aim to bridge this gap in knowledge was by trying to catalog a lot of biodiversity that you see on Red Sea reefs. We did this by hosting a bunch of research cruises up and down the Saudi Arabian coastline, where we invited a whole bunch of taxonomic experts on corals, fish, all other marine invertebrates to come out to Saudi, sample as much as we could, and basically catalog these things using both traditional taxonomic approaches and molecular approaches. So here you can just see on one of those particular cruises the growing pile of voucher coral specimens. Here's team fish up here. There's a cameo appearance of Howard Choate in the back there. He's hiding. And then this is the invert team, basically digging up everything they could from the Red Sea and then going through those material. In order to say something about what's inside the Red Sea, it was also pretty important to know what was just outside of the Red Sea as well. So we lobbied pretty hard to National Geographic to get funding for remote sampling expeditions outside of the Red Sea. So over the past few years, we were able to get up to places like Djibouti, along the coast of Oman, here at the island of Socotra. I'll talk a bit more about that a little later. On the other coast of the Red Sea in Sudan, and as recently as this past December to Somalia. So obviously, a lot of these places are pretty difficult places to get into and work in. You end up putting a lot of trust and responsibility in the hands of local fixers in order to work in these areas. But in my opinion, they host some of the most interesting coral reefs in the world. So I thought I'd run through some of the initial taxonomic findings, some of the more quirky things that we found. Again, there's a lot of material to go through, and we're just starting to get through the outputs from this project. So I thought I'd highlight a few things. They did find a new species of gall crab, now called Fitzgerenia panda, based on the large black eyes on the specimen here. This work was done by Sansia Vandermeij at the Naturalis Biodiversity Center. They also found this new species of symbiotic copepod that forms these galls or cysts within a Stylophora pistillata host. So you can see here how it forms. It causes the tubular growth of corallites here around this living organism. And this work was done by Slava Ivanenko at the University of Moscow. Now for the coral folks in the room, this is kind of a more comprehensive list of some of the new species of corals that they've now described from the Red Sea. You'll notice one of them up here, Cyphastia kausti, is actually named after KAUST itself, based on largely the big financial and logistic investment into the project. There's also some new taxa that have been resurrected from the literature as well. There's many other works in progress here. And really I want to stress that a lot of this work comes from basically combining both the molecular information with the morphological information. And in this case, the morphological information comes from images of the microstructure of the skeleton, so SEM microscopy. Basically, they're combining these two types of information to reevaluate these taxa in the Red Sea. So this is just one example of that morphomolecular approach. So this is one particular species that was known as Micromusa amacucensis. I'm probably butchering that. Essentially, this was one of these widespread species found throughout the Indo-Pacific. A lot of morphological variation across this range. And basically, upon reevaluation, you see they have samples from, shown in green here, basically the Red Sea and Gulf of Aden. And when they compared that to samples in red here from the Pacific, they found big differences in terms of molecular trees and haplotype networks, but also based on the micromorphology as well. So they ended up describing a new species from the southern Red Sea and Gulf of Aden known as Micromusa indiana. Also interesting, it's not only a case of new species being described in the Red Sea with regard to the corals, there's a lot of lumping going on as well. So there's a lot of unexpected logogenetic diversity in the region as well. So these are three major genera, and basically upon reevaluation, for example, in this first case here, there was 9 morphospecies suspected from the Red Sea, and they ended up finding only 5 molecular species from those samples. And then the Red Sea is thought to be a bit of a biodiversity hotspot for Stylophora, and based on reevaluation of the 6 morphospecies in the Red Sea region, they ended up finding 1 molecular putative species. So again, the case for corals is not clear-cut. There's a lot of changing going on in the Red Sea in terms of reevaluating the taxonomy of the groups, but it seems like some progress is being made. Lastly, with regards to the corals, some of the experts in the group figure that they've found a new genera of coral, Oceloporid coral, being named after one of the super enthusiastic invertebrate taxonomists that were on a lot of our cruises. So it's going to be called Gustavia. This is a really interesting group in that they're really small. They're only a few millimeters in size. They're azooxanthellae. They live in dark caves behind big foliaceous coral heads, and so they're one of these really small dwarf corals that love low light conditions. And so this work is being led by Francesca Benzoni, kind of reevaluating this group. So I will mention at this point, but I'm by no means a coral taxonomist or an invertebrate taxonomist. So my main interest in getting involved in these projects was really to look at reef fish diversity within the Red Sea, and particularly addressing some of the broader ecological and evolutionary questions related to the Red Sea itself. It's really had a unique geological history in terms of factors that can influence the evolution of marine life. The most notable one is about 5 million years ago, the Sinai Peninsula uplifted and basically blocked off its connection with the Mediterranean. And it also formed its only connection with the Indian Ocean in the south. And essentially because of all these glaciation cycles, the raising and lowering of sea levels, the Red Sea has been repeatedly isolated from the Indian Ocean at this very narrow and shallow entrance known as the Strait of Bab el-Mandeb. And it's thought that during glacial periods, low sea levels, the Red Sea was either completely dry or so salty, or the level of stable oxygen was so different that it could no longer sustain life. So something like you might see in the Dead Sea today. And it's this isolation that's thought to result in a high rates of endemism. So about 15% of the fishes in the Red Sea are only found in the Red Sea. A lot of these fishes have Indian Ocean counterparts, so sister species that look similar but also look a little bit different. So essentially, my role in the project was setting up the Red Sea reef fishes study design. I was really interested in connectivity between the Red Sea and the adjacent body of water, the Indian Ocean. I wanted to look at connectivity within the Red Sea, and then finally I really wanted to get a kind of an estimate on the age and origin of a lot of these endemic fishes. For this study, we kind of took a multi-taxa approach, so we didn't just focus on a single species. We had a heavy focus on the butterfly fish. This is probably the most interesting reef fish group in the Red Sea. It has the highest rates of endemism of any of the groups. But we also tried to sample representative fishes from other major reef fish families. And a lot of people often ask me, why don't you just pick a single species and use that as your indicator species or representative? Well, the problem with that is that when you're really asking those kind of broad biogeographic questions, usually a single species is not representative of what's going on overall. So it's good to get a kind of wider picture look. So I thought I'd share with you some of the key preliminary findings so far from the reef fish part of the project, which is also a work in progress. One of the main things to come out of the study was that we have a pretty comprehensive tissue library at KAUST at the moment. And a lot of these tissues have associated whole voucher fishes that are available for examination at the California Academy of Sciences. This is work with Dr. Luis Roca, who is one of the curators there. And you can see that we have samples, obviously most represented from Saudi Arabia, but we also have from other countries around the area as well. And we have pretty good representation from a whole bunch of different taxonomic families of reef fishes as well. So one of the key bullet points that we're finding is that even though the level of endemism for fishes in the Red Sea is quite high, it's probably an underestimate. Meaning that as we start to reevaluate some of these fishes, we find that some of them that we thought were once widespread are actually endemic. So I just take this example because this fish is super cute. The chocolate-tipped chromis, this is one of the common fishes that you'll see on the reef crest in the Red Sea. So when we started to look at specimens from the Red Sea and compare them to the Indian Ocean, we saw differences in coloration. We noted marked differences in pectoral fin rays, lateral line scales. These are common diagnostic traits used to classify fishes. And then when we added some of the genetic data in, this all seemed to fit in that these formed these reciprocally monophyletic groups, so they're highly divergent from each other. We ended up calling the Indian Ocean fish a new species, Chromis fieldi, and then the Red Sea form is now endemic to the region. And the more and more we look at more and more fishes, the more and more we find that genetics are helping us to reevaluate some of these taxonomic classifications, and we're discovering kind of more endemic species. Another interesting point I thought I'd share is that when we start to look at some of these widespread species, so looking at populations of the same species in the Red Sea versus the Indian Ocean, we start to date the divergence times between the split between those populations. It seems to be all over the place, but in a lot of the cases it coincides with these big sea level dips, so these glacial cycles. So this is sea level relative to present. You can see big dips below 120 meters about 20,000 years ago, 70,000 years ago, and 125,000 years ago. And so some of the splits between the Red Sea and the Indian Ocean really coincide with these fishes. But much more interesting for us is the older divergence time in these widespread fishes. So this is a squirrel fish and an angelfish. this suggests to us that the Red Sea or Gulf of Aden may act as a refuge during these levels of low sea level, so not all of these populations went extinct. They were able to maintain populations during these times and remain isolated from their Indian Ocean counterparts. Another important point that we're finding is that the Red Sea is not a graveyard for biodiversity. It's a bold statement. Essentially, what a lot of authors had suggested is a lot of these peripheral habitats, so places like Hawaii, East Pacific, Brazilian subprovince, basically a species will get into these areas, evolve into something new, and that's the end of the road. That's the end of that particular species. They won't really leave that area ever again. So what we're finding is at least some of the biodiversity that we're finding in the Red Sea is that actually the original source for some of the Western Indian Ocean fauna. So it's not a case where stuff gets into the Red Sea and stops there. The Red Sea, in some cases, really does seem to be seeding biodiversity into adjacent regions. Another thing that we've noticed is the more and more we look, the more geographic area that we cover, for even some of these widespread species, we start to pick up these cryptic species. So it's not important that you understand the details of these haplotype networks. They just show the relationship between genes based on the different areas where they were sampled. What I want you to get out of this is, for example, this is a widespread Indo-Pacific butterfly fish, Cataloguing melanotus. This is one of the more wider spread tropical Indo-Pacific snappers. And even within the range, we're finding these huge genetic splits. This may not seem like much, but this translates to about 2 to 8% of the CO1 barcoding gene. This is somewhat on the order of what people will use, taxonomists, to describe new species of fish. And if you were to compare these different populations based on the naked eye, they would look very similar. So that's why they're kind of given the name cryptic lineages and cryptic species. Once we start looking within the Red Sea, we start noticing this really consistent split for a lot of the fishes, but also in things as far removed as the sponge, at about 19 degrees latitude. So again, it's not important that you know the details of the structure plot here or the STAPC plot. I just want you to see the shift in genetic data basically at sites sampled above 19 degrees latitude. These are characterized by really nutrient-poor oligotrophic conditions. Once you cross that line, these sites down here are characterized by really nutrient-rich eutrophic conditions. So there's this real shift in conditions within the Red Sea itself. And some groups out at Woods Hole are actually working on the physical and chemical properties of why there might be this break in dispersal within the Red Sea. And they've worked out that at about 19 degrees latitude, there's this real persistent mesoscale eddy system that sits right in the middle. This is largely driven by this Tokara wind jet. So these are winds that flow down from the mountains of Sudan and create this mesoscale eddy system where you then have nutrient-poor north and nutrient-rich south. So it's interesting because we can then start to blend kind of the genetic data with some of these oceanographic data as well. We've also made the push into next generation sequencing techniques. And by one of the studies that just came out on the Red Sea clownfish, we've essentially found that they too show this difference between sites in the north and then sites in the south. And these, based on SNP loci, these are not randomly distributed across the genome. The loci that are really showing differences between the two populations seem to be located at specific places within the genome based on some of these outlier tests. So as the clownfish genomic resources develop, as the genome becomes available, we can then map those back to that genome and figuring out what function those particular genes may serve. And then following up on the genomics theme, we actually made the push to sequence one of the endemic butterfly fishes in the Red Sea, and we've actually now finished this. So the organism that we chose was probably one of the most interesting butterfly fishes there, Catodon austriacus, so the black-tailed butterfly fish. It's endemic to the region. It probably has the smallest range of any of the Red Sea endemics. It's really only found in the northern or central region. For those of you that religiously read molecular ecology resources, you can check it out there. But the genome is also available at this genome browser site. You can actually download the data and kind of how to play with it. So in the end, we ended up doing most of the sequencing at KAUST. We outsourced bioinformatics to a private company, and they were able to filter the data, assemble the data, and then annotate it up to about 29,000 genes using these few available fish genomes that are out there. So things like takafugu or the zebrafish, which is used as a biomedical model. And we're now starting to map some of our SNP loci from all the different butterfly fish back to this genome. So we're using it as a resource. And that's why we put it out there publicly available. We really want others to use this as a resource as well. We encourage others to improve on it, either do more sequencing, transcriptomics, reassemble it using new programs. So it's really there as a tool for anyone to use, and we hope that happens. Lastly, for the Red Sea project, I thought I'd talk about how our exploration of some of these peripheral locations really point out the fact that these are probably some of the most interesting areas to study the origins of biodiversity and the maintenance of biodiversity. So this is building on a lot of work by J.P. Hobbs and a few other people before him, looking at hybrid hotspots in the Indo-Pacific for reef fishes. This figure here is essentially showing, so that the size of these red circles here are the number of butterfish hybrids in a particular location. So these are particularly conspicuous fishes, and it's kind of easy to spot hybrids for this group. And they noticed that these hybrid hotspots really do show up along the edges of these biogeographic borders. So where two biogeographic provinces meet, and you get mingling of the two different fauna. So he's found hybrid hotspots in the eastern Indian Ocean, Indonesia, there's some in the GBR, Japan, up in Hawaii. I put this figure up here just to point out this giant gap in knowledge in this region here, so the big black box. So it's not that there's no hybrids here, we just really don't know if there's any information of that kind on them. And so to follow up on this, we were really lucky to get out to this place a few years ago, the Socotra Archipelago. You can see it's indicated up here. It's partway between Somalia, Yemen, and Oman. A really amazing place. For those of you not familiar with it, it's a UNESCO World Heritage Site. It's usually associated with this unique terrestrial biodiversity. But essentially, we were able to get out there and do some field surveys for a few weeks in a marine setting. And we identified it as one of these marine fish hybrid hotspots. And basically, one of the unique things about this place is the fact that it sits at the edge of so many different biogeographic provinces. So at Socotra, you can expect to see a lot of the Red Sea endemic fish that make it there. A lot of the East African fish make it up there. A lot of these Arabian Sea endemics, Omani endemics, make it down to Socotra. And then you get some of the more widespread Western Indian Ocean fish that make it out there. And the thing is, a lot of these fishes that make it out to Socotra just make it out to Socotra, so they're actually quite rare. And, you know, what do you do if you're a rare fish and you're having trouble finding a mate of your own? You may look for the next best thing, which is something that looks like you, that's quite abundant. And so we basically found quite a bit of mixing going on in this area. We tried to get back there earlier last year, but unfortunately there was a civil war started in Yemen, so it was a bit of a no-go zone for us. It's a really bad situation now. But we were able to get back out to this area down here. This is known as the autonomous area of Somaliland. It's not really considered a country. There's no foreign embassies. Yeah, it was very difficult to pass by my university. But in the end, they let us go. And it was largely funded by Nat Geo and also the Explorers Club gave us some support to go out there. And you'll basically see that it's about 1,000 kilometers to the west of Socotra. And when we actually got in the water and we started to do our surveys, we started to see some hybrid fishes as well. So this is just one example here. This is one of the Red Sea endemic butterfly fishes that makes its way out of the Red Sea to this location. This is one of the more widespread Indo-Pacific ones. We found big mixed schools. It's hard to see here, but there's actually two different species of bannerfish. So we found mixed schools of those, and we sampled some of the hybrids as well. The important point here is that the number of hybrids that we saw at Somaliland was an order of magnitude less than we saw at Socotra. So it really does seem to be a lot of important things about Socotra environmental attributes that really concentrate these hybrid reef fish at this location. And if you're interested in reading a bit more about Somaliland and some of the work up there, there's a blog on the KAUST Academy page you can look up. The last thing I'll say about the Red Sea is I don't have time today to talk about all the projects that our group is working on and a lot of other groups are working on as well. A really good place to start, if you're really interested, is checking out a virtual issue in the journal Biogeography that comes out next month. This was based largely on a workshop that we hosted at KAUST, where we put together a few review papers. There was a number of individual contributions from a lot of people at the workshop or people that couldn't attend the workshop that wanted to contribute as well. So hopefully that's out next month, bundled online, and it'll also highlight a lot of the older papers from Journal of Biogeography that follow that same theme. So it's a good place to go if you're really interested in that topic. So for the last 15, 20 minutes here, I thought I'd switch gears and talk about some of the stuff I'm doing now out at Curtin. A lot of my work is associated with this trace and environmental DNA lab out at Curtin University in Perth. PI for that, or CI for that lab is Mike Bunce. There's a number of academics there, including myself, a bunch of students, and some technicians as well. And actually, this is a really unique facility in that its sole purpose is basically isolating and amplifying DNA from really trace or degraded sources, and in an effort to mitigate basically contamination. So it's a really ultra clean, ultra pure facility. It's a bit of a hassle to go work in these modules. You basically dress up in these suits. There's HEPA filters, one-way airflow, they UV and bleach all the surfaces in this facility. But it's been a great working environment so far. Outside of these modules, we basically have the capacity to do MiSeq and NextSeq Illumina sequencing, so next generation sequencing. The two main approaches that we use are shotgun sequencing, where you're taking a large sample and breaking it up into little fragments and then sequencing each of those, so everything in your sample, or amplicon sequencing or metabarcoding, where you're interested in a particular gene or a few genes within your sample. So you can smush together a lot more samples into a particular run. And the analogy for those of you that aren't familiar with genetics at all is shotgun sequencing is like sequencing a lot of small pieces from a really big puzzle and then putting it back together again, so assembling it. Whereas metabarcoding, you're only amplifying a few small pieces of a very small puzzle. So I thought I'd run through the metabarcoding approach, because I know it may not be familiar to everyone. It's really simple, and I'll go through just the basics, but the first step is isolating some form of DNA, whether it's a water sample, a feces sample, soil, bone, whatever you're working with. You get DNA, and then you amplify that DNA. In this case, it's a bit of a special amplification step, or PCR. You're amplifying it with primers that have these unique tags on them, and you uniquely tag each and every sample we work with, which means you can combine all your samples together, run them on a next-gen sequencer, get millions of sequence reads, and on the back end, you can sort all the samples from each other bioinformatically based on those unique tags that you put on them. Once you clean your data, so you chop off those primer sequences and adapters, you can then query your samples against known public databases. The one that's probably most familiar with people is GenBank, hence GenBank. If you have reference tissue, you can create your own custom library to query against as well. So I thought I'd run through just a few, so this is not all marine stuff, there's some terrestrial stuff here as well. I thought I'd run through a few examples of some of the projects in the lab at the moment that use this metabarcoding workflow, so you can see the diversity of different types of projects going on. So in this particular example, Megan Coughlin is trying to isolate DNA from some of these Chinese herbal medicines that are found throughout a lot of the stores in Australia. And basically by crushing up the pills, amplifying the DNA that's in those pills, she's able to identify what was in those products. And I didn't realize that it was such a contentious issue and such a controversial world, but some of the things that they're finding in these pills is actually quite frightening. You know, through trace contaminants, they're looking at a lot of heavy metals found in some of these products. So if you're interested in reading a bit more about it, you can check out this new paper at KAUST Genetics. I think in some of the samples they found endangered species like snow leopard DNA and those types of things. So it's a really, really strange field. A large part of the lab also works on archaeological samples, so they do a lot of digs in caves. For those of you that recognize the spot of southwestern Australia, this is a lot of the cave systems are between the two capes around Margaret River region. Essentially, they go into these caves and extract layers of bone. And by looking at the different layers of bone, they can tell how fauna has changed through time. So it's kind of an interesting, neat trick. And instead of painstakingly going through each and every one of these bones and trying to figure out what type of animal they belong to, they essentially throw them all in a blender to make this white powder, and then they sequence that. And because they're sequencing millions and millions of reads for a particular run, they're able to pick out most things that were actually in the sample. So it's kind of a quick and dirty way to look at faunal changes through time. And finally, one of the other projects, it's a strategic move by Mike Bunce to get into the wine business. Essentially, they're interested in telling the genomic profiles of wine. So they're deep sequencing wine samples to look at I guess, things that may influence the strain or flavor of particular wines. In truth, they're mostly sequencing the bacterial fauna that make up these flavors. And then the big secret is that, so they have all these vintage bottles. They've partnered with Cullen Estates, which is one of the more fancy, expensive vineyards in Margaret River region. They only need about 1 milliliter to do the whole project. So they have a lot of vouchered bottles sitting around the lab. But it's actually kind of a big field, and they're trying to partner with a few more vineyards in the region. So I've been working with that lab over the past year, a bit more than a year, trying to move some of these metabar coding workflows into the marine sector, let's say. And a lot of this is based on amplifying eDNA. So we started out with some water sampling. A lot of the organisms that you find in the ocean will expel DNA in the form of mucus, skin cells, larvae, whatever you can think of. And it remains viable in the environment for a few weeks to a month or so. And so we've been filtering water and using this apparatus in low micron nylon membranes. To be honest, this has been going on for quite a while in the microbial world. They've been doing this for more than a decade and isolating DNA from the microbes in the water. But we were interested in kind of expanding this a bit more to cover everything from bacteria to mammals, so looking at the whole tree of life in these particular samples. And to do this, we've designed a whole bunch of different primer sets. You notice some of them will amplify basically everything from the tree of life, whereas other ones are more specific to a particular gene in a particular group. So these ones will only amplify 16S in fish, 16S in crustaceans, and so on. So these are some of the pilot data, early trials that we've been working on. Mike Stadt at Curtin's been doing a lot of this work. In this case, this is from a 9-liter water sample from Coral Bay. For those of you not familiar, it's kind of at the southern end of England Reef. And using just those fish 16S primers, he was able to detect 36 taxonomic families of fishes. Thankfully, most of those made sense based on what type of fauna we might expect in the region. So, tropical groupers and butterfly fish and emperor fish. More interestingly, he's recovered some of the cryptic fishes that you may not normally see. You might miss. You might not include them on visual surveys because they're too difficult to kind of pick up. And so he's picking up a lot of these cryptic taxa. When he expands that primer set from the fish primers to that 10 metabar coding primer set, he basically picks up everything in that water sample from the marine tree of life. Not everything, but most things. So you see like rays and more eels and fishes and crustaceans and diatoms and copepods and so on. So it really opens up an avenue where you can query or audit biodiversity from particular samples and take an ecosystem-based management approach for single source samples from particular locations. We've also done some ground truthing with this approach as well. So in this case, we've taken water samples, but we've independently surveyed the exact same places with, for those of you not familiar with RUBs, baited remote underwater videos. Essentially, this is a way to survey fishes. You have a little camera set up, you drop in the water, and there's a little baited arm at the end of it. You record for a certain amount of time, and then you pick up all the different species that show up on your video feed when you analyze it back in the lab. So we essentially wanted to see how well those two methods performed against each other. And we did this in a number of habitats, reef and seagrass. We did a whole bunch of different replicates. We did this, I don't know if you can see this down here. This is Jurien Bay. This is a few hours north of Perth. And there's within this area, there's a few sanctuary zones as well. So there's a lot of replication in the study. And essentially what we found is that both methods are able to tell reef from seagrass signatures based on the fishes that they were picking up. So you'll notice that for the brubs, essentially there was a bunch of fish species that were only found on the reef, a bunch that were only found on the seagrass, and some that were found on both. And eDNA kind of gave that similar picture. A lot more interesting though is when we look at all those data together. Again, here's the separation in brubs on the seagrass and brubs on the reef. You get that separation in the species that are driving the differences between them. And then you get the same separation in eDNA. More importantly here is the big separation between the eDNA datasets and the brub datasets. So there's obviously strengths and weaknesses to both of these types of survey methods. Obviously with brubs, you might expect to see a lot of the larger predatory fishes showing up to nibble on the bait, and those are the things that you're reporting. Whereas with the eDNA, we're picking up a lot of the smaller cryptic fauna that would be missed on the grubs. So they're really actually complementary to each other. You add them together, and you get a somewhat more complete picture. So a lot of this preliminary work kind of fed into this ARC linkage project with BMT Oceanica and the Department of Fisheries, Western Australia. And so I thought I'd share a bit about what they're doing with this right now. So there's 3 separate phases to this project. The first one is they're basically replicating that Jurgen Bay kind of study, but out at a much more diverse tropical environment, and one that's under the jurisdiction of Australian, the Department of Fisheries. And so they've gone out to the Cocos Keeling Islands, which is well to the west of the mainland of Australia, in the middle of the Eastern Indian Ocean. And they've set grubs and collected water to filter at a whole bunch of different sites, different habitats as well. This has a really extensive lagoonal ecosystem, so they've done some work within the lagoon, done some work on the outside reef, and they intend to go back to increase their sampling as well. And there's a PhD student that's actually just started in the lab that's started to work through these data, and real quickly, using those 16S fish primers and 16S crustacean primers, in 14 seawater samples, she was able to pick up a whole bunch of different taxa. You'll notice that these sequence lengths are quite short. Traditionally for taxonomy, you might like something a lot bigger. But when you're working with these degraded materials, if you try to amplify a large product, it simply won't work because it's probably broken up within your sample. So that's why for a lot of these environmental DNA studies, they target these smaller fragments, and you do lose some resolution in terms of taxonomic assignment at that point. It's kind of a trade-off. But anyway, she was able to pick up a whole bunch of different families and genera and species. More important, she started to see, just on those few initial samples, some separation between the different habitat that she was looking at. So she had a few samples from within the lagoon, at the edge of the lagoon, and on the outside pass of the island itself. And so different fishes were showing up in different locations as you might expect. The second phase of this project, it involves more ground truthing, so a lot of aquarium kind of trials. So basically taking seawater in tanks and spiking it with DNA that you know the size of and you know the amount of, and then subjecting it to different temperatures to see how quickly or how slowly that DNA degrades. So they're really interested in applying these eDNA decay models to the studies, to ground truth them as well. A lot of these experiments are happening, small-scale experiments in the field to complement that, and also working with some oceanographers. Yes, you know it. People that understand the currents of the ocean, to basically tell us, if something is shedding a piece of DNA, where would that end up based on the currents and the winds in that particular area. And then the final phase of that project is looking at checking out biodiversity in the Kimberley region. So this is a pretty remote region in northern Australia. It's really rich and diverse. One of the, issues that fisheries are facing is that it's a very difficult place to survey regularly because the water is very turbid, the tides are pretty extreme, and there's also these, as you guys know here, these critters that are kind of lurking in the waters. And so they want to find a quick and easy way to survey biodiversity along this kind of coastline. And so they've gone up there and they're sampling water and they're taking gut contents from a lot of the fishes. And the aim is to audit biodiversity across multiple trophic levels. More importantly for them, they really want to identify those essential fish habitats. So some of the seagrass beds or mangrove areas that serve as nurseries for a lot of the fishes that end up on some of those offshore reefs. So that Kimberley is thought to be one of those particularly important seeding zones for some of these offshore spots. So fisheries are pretty keen on working through this part of the project. Final 2 minutes of the talk, I thought I'd just focus on some of the things we've learned so far and the Trend Lab has learned so far through their work in this eDNA realm. The main 2 things that you have to mitigate in this work is definitely false negatives, false positives. They're both bad. False negatives, essentially, let's say you're surveying an area for a particular invasive species using eDNA approaches, and your assay tells you that it's not there when it is. That obviously would be pretty bad because that defeats the purpose of you going out and sampling for it in the first place. So the main thing that we found to mitigate some of these false negatives is the level of input DNA to any of your workflows is actually really, really, really important. So every single sample that we take, we optimize whatever working dilution of DNA that we're putting into our PCR. For those of you not familiar, this is just a qPCR trace, and this is, you're seeing the amplification of DNA through time, through cycle number. So this is a pretty healthy trace. This is a good working dilution in green. If you don't have enough of your input DNA in your reaction, you can see that the cycle where it takes off is pushed back, and you get less available DNA. And actually, if you put too much DNA in your reaction, you kind of kill the PCR. And in that case, if you were doing that for a lot of your samples, you may get a negative result when in fact something may be there. You just wouldn't know. Much more important and harder to mitigate are the false positives. So you can imagine, again, if you're looking for an invasive species and you detect this invasive species but it's not actually there, and then you give that information to some government agency that rolls out an entire multi-million-dollar eradication program based on false data, that would be bad as well. So some ways to mitigate the false positives is that we are standardizing our protocols in the field. Everything must be in kind of the most clean, sterile setting that you can possibly achieve in the field. Bleach is your best friend for these types of things. We bleach everything. We bleach all our instruments. We bleach countertops. We bleach bottles. We bleach hands. It's really one of the few things that will get rid of extraneous DNA. Another thing that we've been doing is those tags that they said that you can tag a sample with in PCR. Once we use a combination of tags, we never use that combination ever again. Because if you pick that up a few weeks or a few months later on another sequencing run, you know that some of these, there's a level of contamination that's been transferred across different runs, which is bad. Single-step PCR just means you reduce the level of bias, you reduce the level of handling. It's very easy to transfer things between tubes just by touching it with your gloves. Extractions, negative controls. So if you start to see proper products showing up in your negative controls, then you know there's an issue with contamination. And finally, it's been a real great opportunity to be able to use these ultra-clean laboratories. So it puts your mind at ease that a certain level of the workflow is kind of done in this sterile setting. So I think I'll end there. There's a lot of different collaborators to thank for the Red Sea project and from some of the work we're doing at Curtin now. The Trend Lab is really open to working with anyone if they're interested in kind of rolling out these eDNA methods in the marine sector. They have a strong molecular background and they're looking to work with people that have really strong marine backgrounds, whether it's generating pilot data, so you can see whether the methods that they're using actually answer the questions that you're interested in, or maybe it doesn't. So they're really interested in discussions of that nature, and a lot of funding sources to thank as well. And yeah, if there's time, I'm happy to take a few questions. Thank you.